Welcome to scverse meeting, community meetings of Tuesdays. So I'm very happy today. We have Geert, and I'm sorry if I pronounced it wrong. Perfect. Yes, that he will talk to us about an amazing project that has an amazing name, in my opinion. It's called Mowgli, right? From from the movie. Yes, that is for basically data integration. And I'm sorry if I'm wrong. I briefly read about it. Method for integration of paired multi-omics data. So yeah, we're looking for your presentation. Please, you can share the slides if you want.
Thanks a lot for the introduction. Can you guys hear me all right? Okay, I'll share my screen. Okay, can you see my slides?
Yes, perfect.
Good. Thanks again for having me today. It's very nice to be able to present in front of this community. And as I'll talk a bit about later, I'm reusing all the tools that scverse provides. So I'm very happy to be able to share this with all of you. So my name is Geert-Jan Huizing. I'm a PhD student at the ENS-PSL and at the Institut Pasteur under supervision of Lara Canti and Gabriel Perret. Today, I'll introduce our new tool for single-cell multi-omics integration called Mowgli, and it uses optimal transport. I think most of you are familiar with single-cell omics, but you might not have used single-cell multi-omics before. The single-cell multi-omics are a set of technologies like 10x multiome or CytSeq that allow you to measure the cell at several layers of regulation at the same time. So for example, CytSeq gives you access to the transcriptome, but also surface proteins. And the next multiome gives you access to the transcriptome, but also to chromatin accessibility for the same cell. So you get several pieces of the single cell puzzle at once. And of course, this has a great impact on understanding of the immune system, of development, and of complex diseases like cancer. And to give you an idea of how popular single-cell multi-omics are getting, I plotted the number of publications of PubMed across the years with the keywords single-cell multi-omics. And you can see that we're really entering the multi-omics age. Another way to assess the popularity of single-cell multi-omics is to just have a look at the diversity of methods that exist. And this is a set of different technologies that were developed in the last few years. And you can see that it's fair to say a new one pops up every few weeks. So this is a very fast pace of biological development. And actually, we need to keep up with the technology development on the computational side. So we need computational methods that are adapted to this kind of data and that are able to take advantage of the complementary information that single-cell multi-omics offers us and to use it to analyze the data. So these are called paired single-cell multi-omics integration methods, as opposed to unpaired, where you would have, say, RNA-seq on one side and ATAC-seq on one side, but profiled from different cells. Here we focus on the case where we have several omics for the same cell. There's a bunch of methods to analyze this kind of data, and they're broadly subdivided in two categories. First of all, linear methods that are simple, but their good side is that they're interpretable, because their factors, so their latent dimensions, map right back to biology, to genes for RNA, to peaks for ATAC-seq, to proteins, and so on. So for this, you'd have joint MMF, you'd have MOFA, and so on. And on the other side, you have a class of new methods that rely on deep learning, that are more expressive, but you pay a price in terms of interpretability, because you don't have a linear mapping back to the space of genes or peaks and so on. So you have multigrades, cobalt, and a bunch of architectures that are built for paired data like this. So what we're doing here is that we're proposing a method that is a linear method. So we have the advantage of interpretability, but it's powerful, and I'll explain you how we use optimal transport to make it a bit more expressive than traditional linear methods. The good thing about Mowgli is that it's compatible with any type and number of omics, because we don't have any statistical prior on the data that we expect. So we don't have any restrictions on the kind of data that we can take as an input. And the reason I'm speaking here today is because Mowgli is really integrated into the scverse ecosystem. We use mu data as input-outputs, and we use ScanPy and Muon for everything that's downstream analysis, like clustering, visualization, and also biological interpretation. And in my last slide, I'll go a bit more into the software of this and how we actually use these objects in the code. So what Mowgli does is, in essence, a joint non-negative matrix factorization. So it decomposes each cell into a combination of biological signatures, and this across omics. So you have omics-specific biological signatures times a shared embedding of the cells that will be low-dimensional, and importantly, that will contain information that is shared in all omics, or that's and it's only in one of the omics, but at least it's some information that will present all the omics at the same time. And this is not a new model. There's plenty of variations on MMF out there. So what makes us special is the way that we compare our data, our single-cell multi-omics profile, and the reconstruction, so the signatures and the embeddings. And before I get into what we do differently, let me first complain a bit about the traditional way to do it. So traditional similarity metrics, like the Euclidean distance, are not really fit for a single cell. Let me explain myself. So to train a machine learning model, you need some notion of similarity or distance between your samples. And typically for an MF, this would be the Euclidean distance or the KL divergence or something. But we know that single cell data is very sparse, in part due to dropouts, which can trick the Euclidean distance or similar bin-to-bin distances into thinking that similar cells are actually different. Because if you have a marker for a certain cell type that is absent, whereas it should be expressed, then you might think the cell is of a different cell type than other cells that do express this marker. But actually, if you looked at the context, for example, other markers of the same cell type that are highly expressed, then you would know that this is just noise, just a technical dropout. But the Euclidean distance and other bin-to-bin distances consider each feature independently, so you don't have this notion of relationships between features. So how can we do better? And this is where optimal transport comes in. Optimal transport was introduced in the 18th century, originally as the physical effort needed to move around piles of Earth to a different configuration. Now, this seems like a very 18th century problem to have, moving around Earth, but it has actually been very useful in modern computing science as well, because it defines a more general notion of distance between probability distributions, as the effort needed to move around mass from one distribution to the configuration of the other distribution. And this is written as follows. It is the minimum cost of moving around this mass, knowing that you pay a bit of penalty every time that you move in between two features. Now, optimal transport has already been used in biology. And one very notable example of this is Schebinger, where they track cells across time. They try to predict the position of cells across time. There's also more recent work, Muscat. I know that Dominic is in the call, and it's a great work that also uses optimal transports. But this is a different way than what I'm going to present today, because these work focus mainly on transporting populations of cells to other populations of cells. And here I want to talk about optimal transport as a way to compare pairs of cells. So seeing one cell as a distribution on the space of genes, on the space of peaks, on the space of proteins, and transporting that information to another cell. And how much effort is needed to transform one cell into another is the distance between those cells. And as we showed in prior work, our approach is robust to dropouts because it considers relationships between genes. You're allowed to move around gene expression from one gene to another, as long as you pay some cost. So if genes are very different and that you absolutely don't want to move around between them, then you'll just have a high cost this will not happen. But if two genes are markers for the same cell type, then you might want to have a low cost and allow yourself to see them as more or less interchangeable. And so this gives us a fancy new way to compare cells that takes into account relationships between genes. And let's test this on some data. For that, we will need to use optimal transport as a reconstruction loss, as I was saying before. One small detail here is that we're using entropy regularized optimal transport. So this is an approximation of the problem I just described that is much faster to compute. It's compatible with GPUs, and it gives you a fast approximation of optimal transport. And the other term that we have here is a sparsity-inducing regularization, both on our signatures and on our embeddings. And this allows us to have sparse representations of biological signal, meaning that you'll have a few markers that are specific of a certain signature, but not half of your genes. Now let's test it on some data. First, what we did is to generate some controlled settings, so some simulated data, but based on real cell lines data. The reason we start from real data is to not have any statistical assumption on the distribution of the data. So we start with some cell lines data with three cell lines, three groups, and we obfuscate it in a way that illustrates challenges that are typical of single cell data. So we have sparsity and so on. And here I'm presenting complementary information. So on the RNA side, if you only look at that, you'll be able to distinguish two populations. And if you only look at the ATAC side, you'll be able to distinguish two populations as well. But if you mix the two together, you should be able to distinguish that there's actually three populations. And as you can see, the embedding that I plot on the right in the UMAP shows that Mowgli manages to distinguish these three populations. And on all six controlled settings that we have, so complementary information, rare population, sparsity, so on, Mowgli outperforms the methods that we benchmarked according to three metrics. So the silhouette score, which measures if cells that are from the same population, from the same group, are close together and far from cells that are in different groups, far in our latent space. Then the adjusted rand index, which is a measure of how well our clustering is. And here we get 100% clustering. And purity score, which is a measure of, for each cell, are its neighbors in of the same cell type or not? So does your latent space correctly separate cells that are from different populations? And all of these metrics we do quite well. Now moving to real complex data this time. Here we are competitive with the other methods, but none of the methods, surprisingly, really stands out compared to the others. Here I'm showing an example where we did quite well. This is the Open Problems SAT-seq dataset, where this time we have RNA and proteins. And you can see that we distinguish some populations well, and some other methods don't, but we don't perform best on clustering, for example. And part of the reason for this, the fact that all methods seem to perform more or less the same across data sets, across tissues and so on, could be that it's just quite hard to benchmark on real data. For example, the annotation is always to some extent computationally derived, which can be an issue. And sometimes cells from within one cell type can have more heterogeneity than there is heterogeneity between two cell types that are annotated differently. And these things make our clustering metrics and so on a bit less reliable. What is true is that we are at least competitive with other methods. And one other way to sell Mowgli is not only on the clustering aspect, but also on the interpretability aspect. And for this, I'm going to showcase a dataset with 3 omics. So you see, I'm trying to vary the different technologies that I'm using as test cases to show that we're able to analyze big diversity of technologies. This is T-seq data. We have RNA, so transcriptome, you have ATAC, chromatin accessibility, and you have proteins, surface proteins. You can already see that Mowgli's representation manages to separate correctly the different cell types that you'd expect in PBMC data. You have your CD4 cells, CD8 T cells, B cells, and so on. But what's interesting is that if you compare it to other linear methods, and that you look at each factor independently, so each of your latent dimensions, and that you plot how much it is active in one cell type. Here I'm showing CD8 T cells, versus how much it's active in the rest of the cell types. In Mowgli, you'll see that some factors, a few factors, are very specific of that given population, but it's not the case in MoFA or in NMF, regular NMF. So it shows that Mowgli really has some very specific factors that are specific to a cell type. This, of course, is great for biology because you can then go and interpret the signatures associated to that factor and try to make sense of it biologically. You might think that this is limited to broad cell types like CD8, but actually you can also look at very specific cell types. Example here, factor 49 associated to effector memory CD8 T cells. What I'm plotting here is a UMAP of the cells, with the color being how much the weight of that factor. So factor 49 is only active in those effector memory CD8 T cells. And how do we know that those are effector memory CD8 T cells, or non-classical monocytes versus classical monocytes for another factor? Well, the good thing is that since it's a linear method, you can look at the signatures and really interpret them to get some certainty into what it represents. So for this example of the 49th factor, you can look at the top proteins on the lower row and see that some of them are markers of effector memory CD8 T cells. Or same for the top genes, you'll have some markers as well. And if you look at the top genes, you can also use them to do gene set enrichment analysis. And this will cover gene sets that are characteristic of this cell type as well. Another interesting thing is that if you take the ATAC, the current inexhibitability part, you can do motif enrichment, and you get some of the top motifs. And actually, those TFs target the top genes in that same factor. So it's interesting because you really get some cross-layer information by doing this type of analysis, and it gives you more confidence that you are indeed looking at the right population. Hopefully, this gives you motivation to try it out for yourself in your own data. And if you want to do that, well, it's very easy because it just uses all the objects you already know. So we use new data as inputs, meaning that you can have different modalities, your RNA, your ATAC, your protein inside of your data, and just feed that to Mowgli, which then will do its thing. It will compute the embedding, and we'll put that in your OBSM fields, and we'll put the loadings into your unstructured field. And once you have your embeddings in your OBSM, you can use them as you would a PCA in ScanPy. So you can do your UMAP, you can do a Leiden, and then for biological interpretation, we have some of our own functions, but you can also use the standard things in ScanPy, like enrich to do gene set enrichment. If you want to know more about it, of course, ask me questions about this, but there's also a preprint, and it's very easy to install, just to pip install Mowgli. If you have any issues with or questions about the package, just put an issue in GitHub. With this, I would like to thank my supervisors, Laura and Gabrielle, and also Ina, that helped me with this work, as well as everyone who gave me feedbacks along the way in the lab and outside. And thank you very much for having me.
Thank you so much for the presentation. Let's put a virtual clap on the Zoom. So it was a great presentation. Let's go through the discussion. You can raise the hands if you want, and I can say your name. And also, if you want to address first the comment that it was on chat, it asks about the silhouette score, and if you can explain the figure, so we can start from that, and then we can go through.
Okay, so I don't see the chat from where I am. Could you remind me what the question was?
So what is the silhouette score? I'm sorry, I'm not familiar with it. So yeah, there you show the silhouette score. Yeah. Exactly.
Yeah, yes. So the CUS score is really quite simple. Here we have 3 cell lines, 3 groups. And the CUS score tells you, the CUS score is big if cells that are from the same group are closer together than cells from different groups. So it will be maximal if you have just 3 direct distributions, 3 points for one for each group. And it will be minimal if everything is just mixed together into one big blob. So it assesses how well you can separate different cell types. And then the bad thing about CUS score is that if there's a lot of heterogeneity within one group, it will penalize that. So it kind of has the assumption that the cell types that you have don't contain much heterogeneity within them.
Cool.
Thank you.
You can raise the hands or just write on the chat if you don't feel comfortable with that, and I can always read it if you have more questions. Yes, Isaac. Sorry, I skipped Gregor. Sorry, Isaac, you're the second. Wait, Gregor, you're first.
Okay, yeah, you mentioned that optimal transport is nice to compute the distance between cells because you don't have the problem with the sparsity. So I was wondering if this would be applicable beyond Mowgli, but maybe just as a metric to compute a neighborhood graph for single cell analysis.
Yes, so actually this is a paper that I started working on during my internship and that we published last year, which looks at exactly that. So computing distances between cells in different modalities, RNA, but also ATAC, and I think we had DNA methylation, and comparing that to what you would get doing your KNN with Euclidean distance. And we see that it seems to be more robust to dropouts, and it seems to give some interesting results in terms of clustering. Now, the negative point of this is that it requires to compute n squared, if you have n cells, n squared optimal transport distances. And even with the entropic regularization I described in this slide, optimal transport is still much slower to compute than an Euclidean distance. And you don't have all those fast approximate nearest neighbors algorithms that you can actually use in practice for big data sets. So it would be very interesting to have something like that, although I don't think it scales as much if you need to compute distances on say, atlases, for example.
But how well does it scale? Like, is it reasonable to do this on, say, 100,000 cells, 500,000 cells?
So I would say at 100,000 cells would be very, very slow because you have to compute those n squared distances. And with the Euclidean distance, you have some tricks to make it faster, but here you just have to compute n squared. With Mowgli, it's not the case. With Mowgli, you scale quite well because you're only comparing one cell to its reconstruction. So you're not computing n squared distances, just n, one for each cell.
Okay, thanks.
Welcome.
Isaac?
Yeah, I had a question about the integration benchmarking part. Well, so first I also wanted to say really nice presentation and really good graphics too.
Thank you.
For the integration benchmark, do you think that there is a better kind of way that we can be comparing these methods?
I mean, it's difficult. Like one problem being, as I mentioned, that first of all, the annotation is computationally derived. And even if you have some expert knowledge in it, and I know that Open Problems is very good with in this regard, very well curated and a lot of work put into it. But still, at some point, there might be a bias in the way that this annotation is done because you still have your KNN, kind of a Seurat type analysis. So if a method does something drastically different than Seurat, so it doesn't do a KNN and then integrate two graphs together, you might just have a different embedding, but not different biological results or different biological information. So first of all, that can be a bias. And with how we could do it better, I think it's quite difficult. Of course, these benchmarks are good to see if your method is performing very poorly, if it doesn't work at all. But then there's a lot of gray zones in this, I think.
Yeah, I've just one small follow-up on this. Do you think it would be better to benchmark instead of the embeddings themselves, like the downstream tasks? Like can you find regulation that's happening, something like that?
Yes, so here, for example, the adjusted rand index, it's a clustering task. So I do clustering, I take the embedding and compute the KNN and clustering on it for different methods. I do it for MOFA, for NMF, for multigrade, for cobalt, and so on. But that also has its drawbacks because it really depends on the granularity of the annotation. And you might overcluster something in some areas, but also some annotation might be very fine. So you need to have a big resolution to be able to dissociate them. For sure, the downstream task is a more interesting thing than the embeddings themselves, because that's what people want to do in the end, right? They want to cluster, they want to visualize, and so on.
Zale, you can tell us your name as well, because for next time to pronounce it correctly.
Hello, so I just wanted to ask a question about first of all, my name is Charlotte.
I'm sorry, I have trouble hearing you.
Oh, you cannot hear me?
Now a bit better.
Okay. Oh, sorry. Is it better now?
Oh, much better.
Okay. So my name is Jale, and my question is that why, like the performance, or like what do you think, like why it happened that the performance of your method dropped when you applied it to real data, because it was outperforming everything in the previous assessment that you did with, I don't know if it was simulated data or what, but then with this assessment, it is still competitive, but not as outperforming as it was in your simulations.
Yes, that's a very good point. I think the controlled settings play an important role. It's a different way to benchmark the things because you can really look into specific problems and see what impact they have. So it's an ablation study. Instead of taking the whole complicated problem, you just look at what happens if two modalities mix two groups together, but the other modality doesn't. And you expect that a good method should be able to separate that. So on those individual tasks, we seem to be performing quite well. Then real data, of course, has a lot of different moving parts, and I think it's just not as well posed of a problem. And also, we probably pay some price for having a linear method instead of maybe a deep architecture. Although deep learning methods don't necessarily outperform, say, MOFA or NMF either. So I think it's really that's real data are complicated to benchmark properly.
Great. Any more questions or clarifications? Yes, Isaac.
One more question, which I think is always my question about NMF, which is how do you figure out which ones of your embeddings are useful? And also, what's the consequence of setting your number of dimensions too high when you're generating your NMF?
Yes, that's a good point. So for some context, if you have, let me get back to this. So Isaac's question refers to if you have more dimensions here, you might split your information into several small bits rather than have one signature that contains this information together. Whereas with PCA, since it's a different way of constructing it, it doesn't really matter where how much you take, because the first ones are always more informative. With NMF, it's not the case, so there's some trial and error. You have to try and see how many factors correctly represent your data. For example, with the cell lines, there's 3 groups, so we expect that a few factors should be sufficient. You would take 5 and it works. But if you took 100, it would be really overkill because you don't expect 100 biological signatures. It's really quite stable. So overall, if we take 50, 30, 100, it doesn't change results much. And one other way you can play on it is by adjusting the sparsity. So this is what I'm defining here. Those parameters rho and mu are actually, when you look at the dual problem of this, they parameterize a softmax function, meaning that the smaller rho or mu, the more sparse your vector is. Meaning that if you want very, very specific signatures, you can also set this to a big number of factors and ensure that they're very sparse by having a small parameter here, so big sparsity. And if you want things that are really quite broad, like you want 3 signatures that correspond to 3 cell lines, then you'd have not much sparsity, so a bigger parameter here and less factors. So there's 2 things that kind of play together, and that can allow you to compensate a bit if you're not sure that if one of them is not good.
No, I think just as a quick thing there, do you need to do a lot of parameter searching to figure out like how many dimensions, and also particularly your regularization coefficients? Or do you just go default settings tend to be pretty stable? Your results tend to be pretty stable with respect to what you're choosing for coefficients?
So the defaults should be very good. Of course, your mileage might vary, especially if you have some exotic data and so on, which is why it's always good to have some knobs that you can turn. Default parameters should be good. Of course, I did a lot of parameter searching to demonstrate that it was very robust. One area where you might want to play with it is if you're trying to interpret the results. So if your clustering and so on will not change much, but if you're trying to look at one particular factor and you see that it's not as specific as you would want, you would expect really 2 markers that have contain most of the information for that factor, then you might want to put more sparsity. So I'd say that if you're looking at really the biological interpretation, you can play with it a bit more than if it's just really clustering, where the default setting should be just fine.
Thanks.
Welcome.
Jale?
Thank you. I just had 2 other questions. One is why Surat was always dropped at like comparison when in the silhouette scores. Like, was it always really 0 or?
No, the one that is really 0 in this particular plot is Cobalt, which mysteriously just really performs bad on this kind of control settings, whereas multigrade, which is a similar type of deep learning methods, performs quite well. So it might be some thing of statistical modeling or something, I don't know. But to come back to your question on Sura, the reason we don't have CRS score for it is because the CRS score is computed on an embedding on some low-dimensional representation of your data. And Sura doesn't have that. It has, of course, with k nearest neighbor graphs. So you can still compute the adjusted rand index because you do your clustering on the KNN, the purity score because they're neighbors in the KNN, but not the CRS score because those are supposed to be distances in your latent space, which you don't have for Sura.
Okay, makes sense. And my other question was that how was your method compared to the others in terms of timing of the, running the method?
Yes, very good question. I should have mentioned that in the slides. It's a bit slower. The reason for that is that you are taking into account interactions between genes. So you have this notion of pairwise comparisons between features. It's a bit slower, but not much. So, for example, with the biggest data set that we analyzed, which is a bone marrow size-seq data set, it runs in maybe 30 minutes, whereas multigrades would run in 15. It's off the top of my head, but it's sort of magnitude. It's not prohibitively slow. And it really depends on the number of features that you have. So for example, the data set with the most cells, 30,000 site-seq, is actually faster to run than a smaller data set, but that has more features. So with 8x-seq, for example, you're using peaks, and you need much more peaks than you have proteins in site-seq, for example. And since we're looking at pairwise comparison of features, that runs slower than the one with more cells. Is that clear?
I don't see any response.
Yeah, it was clear. Thank you.
Okay.
There is one question also that I will read in the chat, and forgive me if I don't say it very good. So in your NMF, you showed that the more interpretable programs were ones that had cell type specificity. But is Mowgli able to find factors that are common to few subsets of cell types in the overall data set? Like if there is a cell process A factor, and there are 4 out of 12 cell types in the data set whose ground truth is high for cell process A, how would you distinguish that factor between meaningful and non-meaningful?
Yes, that's a great question. In this particular example, we have PBMC data, and we also have proteins as features. The proteins that were chosen in the panel are very representative of the different cell types and subtypes that you'd expect. So often what we see is that one signature will be very associated to one protein. For example, here you have the C56 bright NK cells, which will be very associated to C56 in terms of proteins. Most of the weight in that signature will be C56, will be driven by that. And then in terms of genes, you'll have a few genes that are markers of this population and so on. So in this particular example, we see more cell type specific things than process specific things, although that could vary in other kinds of data sets, so they may be a bit more subtle than PBMC. But for example, we have, maybe this is an example of factors associated with C45RA, which in T cells is a marker of naive versus memory. And this factor is also active in B cells, but then it represents another distinction between memory and naive. So I think that would be more of an example that you mentioned, where it's associated with a certain process, or here a certain protein, rather than one cell type, and it may be shared between two different cell types, but have different meaning. So you need to look at several factors at the same time, and also, of course, at the raw gene expression or proteins in your data to get the full picture.
Good. If there is a follow-up in the chat, I will let you know.
Okay.
So meanwhile, is there any other question or remark or comment? If not, I can wrap up, I guess. It was a very nice presentation, I have to say, one more time, and thank you so much for giving it.
Thank you very much for having me and for the great questions.
Thank you so much for attending it as well. And unless there is something else that you would like to discuss, and we can stop recording.